Good morning, everybody. My name is David Sepanek. I am the head of business development and public affairs with Medicon Valley Alliance. It's 10 o'clock, at least here in Copenhagen, 9 o'clock in the UK, and it's time for us to get started. And on behalf of the Danish-Swedish Life Science Cluster Organization, Medicon Valley Alliance, it's a pleasure for me to welcome members, potential members, to this inspirational webinar co-organized with our member company, Unilabs. We have two excellent speakers lined up for you this morning, Colin Farrell and Anders Elleby, who will be properly introduced by Dave Shelton from Unilabs in a minute. So I hope you have fastened your seatbelts and will enjoy the ride. And if you get inspired not only by the content of today's presentation, but also by the concept, and start to wonder if setting up such a webinar with Medicon Valley Alliance could be of interest to your company, feel free to reach out to me. My contact info will appear on the goodbye slide at the end of this webinar. I also encourage you to visit our webpage, mva.org. And the network section in particular, to see if there are any of the Medicon Valley Alliance networks that could be of interest to you as well. And remember, as an employee with an MVA member company or organization, you can actually attend all the network meetings that you like, free of charge that is included in the membership. A few practical remarks, our household rules, so to speak. It's pretty much a standard operating procedure, but as you can see, you are automatically muted. The chat is for technical questions only, and you should use the Q&A session for questions to our speakers. And of course, be aware, as you can see here, that the event is being recorded. I guess that's about it. Enough said on my part. I'm not the reason why you tuned in this morning. So let's get started. And without further ado, I will hand over to Dale from Unilabs. The floor, or in this case, the screen, is yours.
Thank you very much, David. Hi, everyone. Thank you very much for joining our seminar today, our webinar today. This presentation is brought to you by Unilabs Serving Pharma. And I'd like to take this opportunity to very quickly introduce you to Unilabs Serving Pharma and our services. Next slide, please. Unilabs Serving Pharma is a specialty lab analysis CRO. We are located in the center of Copenhagen on Østerbro, and we work with a network of centers of excellence with specialty lab services across Europe. Our services include metabolism, bioanalysis by either mass spec, immunoassays, central lab, molecular diagnostics, genomic services, flow cytometry, histopathology, and medical imaging. In this setup, we can provide you with a modular solution to suit any particular clinical trial needs. With this setup, we've been able to service over 4,000 clinical trials to date in the 40 years of Unilabs. Next slide, please. Our lab services are delivered in a seamless package with our auxiliary services, including collection kit manufacture here in Copenhagen, our dedicated project management team ensuring smooth running of any project, logistics for ensuring that samples get to and from where they are, and of course the important data services for reporting. If you would like to know more about today's webinar or any of these services, please contact us at servingpharma.com or directly via our LinkedIn or email page. Without further ado now, I would like to introduce you to our first speaker. Our first speaker for today is Anders Elleby. Anders Elleby is the Nordic application manager for Mesoscale Diagnostics. Anders comes here with a strong background and experience in biomarker analysis. At MSD, Anders is the go-to sparring partner for pharma and biotech companies in Denmark, and there to help you with designing the best application or best platform for your Mesoscale application. Anyway, without further ado, I'm going to pass it across to you now, Anders.
Thank you very much. And you should see my screen now. So thank you very much to MBA for the introduction, and also to Unilabs for the opportunity to present advances in multiplex ECL detection of protein biomarkers. So my name is Anders Elleby, and I am account manager and responsible for MSD here in Denmark. I work with clients in academia and also in hospital, health research, and also in biotech and pharma, and discuss various biomarker application and assays. As mentioned by Dale, MSD, the MSD platform represents one platform of multiple platforms at Unilabs. And normally when we give presentation, we usually address specific disease area of biomarker focus. So the challenge for today is that I do not know the specific disease area you are involved in. Therefore, the aim of this presentation is to give you an overview of the MSD-ECL technology, and along with recent development, an idea how we can support implementation of biomarkers for clinical trials for you to consider in current or future trials. It is important for me to say that the MSD platform is an open platform, and our aim is to provide tools to support studies from very early discovery all the way to clinical trials across different species, from mouse, non-human primate, and human. And as mentioned initially, we will take questions in a Q&A towards the end, and you're welcome to write your questions in the Q&A chatter on your screen. So MSD is established in labs all over the world. I am part of the commercial team in the Nordics, and we have other support functions locally with very experienced field application scientists that also go in discussions about biomarkers with you. We also have service engineers, and we have local support functions. And if we look at the Nordic MSD landscape, you can see that there's about 100 instruments in the region, and that there's almost an equal distribution of instruments between academia and biotech pharma. Highest density of instrument is actually here in the Medicon Valley area. Although it is an open platform, we have more than 500 assays providing solutions within a broad range of biological applications. Historically, and for the last 15 years, we have provided assays to measure inflammation markers and assays associated to oncology, cancer, neuro, and metabolic diseases. We are, however, continuously expanding our portfolio with different assay formats within these and also other areas. For example, serology for vaccine development. Now we all hopefully are approaching an end to the COVID-19 pandemic with more and more vaccinated people. But to give you an example, we have since March last year involved in developing assays related to SARS-CoV-2. From preclinical through different phases, we have supported most vaccine companies with multiplex assays for both serology and neutralization for the vaccine production. And today, as we speak, actually also support a phase 4 study running here in Denmark called INFORCE, a national cohort study of effectiveness and safety of SARS-CoV-2 vaccines that hopefully will prove that the vaccines will have their effect in years to come. It is out of scope for this webinar, but I mentioned this just to give you some perspectives and an idea how fast assays can be developed and validated on the MSD platform. The platform itself is based on ECL, And the following slides will briefly introduce the principle of the assay. It is a proprietary detection method shared by MSD, the plate-based version, and Roche, the flow-based version. And it provides a series of advantages in terms of dynamic range, sensitivity, compatibility with different matrices, and instruments that allow for multiplex detection. The instruments are based on a CCD imaging system, and it is a non-fluidic system. That means that there's no contact between the sample and the instrument. With biotech and pharma, we see different models for collaboration. Most biotech and pharma has their own instrumentation, but we also have collaboration with smaller biotech and pharma companies, maybe located at a university where they have access to an instrument in academic setting for later in the process to outsource to SEO, for example, Unilabs. As mentioned, we offer a very large portfolio of biomarker kits commercially available, and in addition, we offer a range of tools to develop own and novel biomarker assays. The core technology is electrochemiluminescence. The light is generated through a redox cycle that is initiated by a current added to the carbon plate. The stimulation is decoupled from the signal, and therefore there's no autofluorescence in the system. Also, it is a proximity assay that allows only signal at the very bottom. This method allows us to build robust assays with low background, reduced matrix effect, and in general, our assay have high sensitivity and a wide dynamic range, as shown for TNF-alpha here, where we can measure both native concentration and diseased or elevated concentration. Most assays on our platform is built as a sandwich immunoassay format, but the method actually also allows, as shown here in the circle, but it also allows for measurements of, for example, extracellular vesicles, as shown here, immunogenicity in a direct format, shown here, or also within antigen oligotherapies, measure also that we now see a requirement to build high sensitive assays towards. It is a flexible format, so also membranes and whole cell can actually be coated on our plates. So you may be interested in a wide variety of sample matrices for your particular biomarkers. Just to give you an idea, our assays is validated for most part for serum and plasma, CSF and cell culture circulatants, but our assays are compatible with almost all matrices from different tissues to tiers. And from a biobank perspective, it is maybe interesting for you to know that they also are compatible with, for example, dried blood spots. These benefits is relevant for single biomarkers, but it can, in addition, run for many markers in a multiplex setting. The principle of multiplex shown here, and we simply allow multiple spots within each well to be coded. For example, with individual capture antibodies. Although the technology allows for more assays per plate, we have limited the number of assays per well to 10. In order to maintain high quality and not to compromise with sensitivity and cross-reactivity. For larger screening panels, we will use wells, and in that regard, it is important to say that the reading time per plate is only 1 minute, and we use a relatively low volume for our samples, ranging from 1 microliter to 10 to 25 microliters. This slide shows data for a pro-inflammatory panel on normal serum samples. The dynamic range is 3 to 4 logs, and detection limit is between 0.1 picogram per milliliter and 10 picogram per milliliter. To the left, you will find typical standard curves, and to the right, an overview of different assays in this particular multiplex in normal serum samples. In some studies, even higher sensitivity is required. Some of them show low concentration, for example, IL-1 beta, IL-2, and IL-4, as you can see here. But I'll get back to that later in this presentation and show you a solution for that as well. We do have different instruments as shown here. Typically, CRO and pharma will have one of these or these two models. They have different capabilities, but they have the same performance, and they can both be implemented in a regulated environment that support clinical trials. Again, there's no contact between sample and instrument, so the maintenance of the instrument is very low. And that is actually the few slides I want to share about the technology and instrumentation. And normally I would now focus on some specific biomarkers or application. But as you can see from this list, we have approximately, it's not meant for you to read, but I will mention that for many of these unique biomarkers, we have multiple formats, multiple species, and the assay can be analyzed in multiple combinations or as a singleplex assay. And this list is continuously growing. Furthermore, this is list only have the commercially available assays, and we often have collaboration that involve novel biomarkers or pharma that use the platform for other areas like PK, immunogenicity, potency, or as mentioned just before, serology assay, to just mention a few examples. That said, I believe it makes sense to give you an overview of the different formats. So we currently have 5 different formats for biomarker assays. I want to give you an overview of these and a few words on how they differ. Importantly, I will mention that all assays are built in the same overall ECL format that use the same plates, and can be analyzed on the very same instrument. Rplex, to the left here, is our most validated format in a fixed and pre-configured panels. There are currently about 100 different analytes, and they are widely used from discovery to clinical trials. The fixed panels does not always meet every demand for biomarker combination, and we have therefore developed a Uplex. This Uplex is a flexible format that allow you to create your own multiplex from more than 200 different assays. Still, there are some demands to allow more assays easily implemented onto the MSD platform, and for that we have launched the Rplex. This is a large and growing portfolio of matched antibody pairs and calibrators for direct use on our plates and ready to multiplex. As mentioned, some situations require even higher sensitivity, and for that we have developed Splex to meet that requirement in the femtogram per milliliter level. The commercially available list is small, but a large number of custom Splex have been developed for pharma over the last 5 years. We are also moving into the nucleic acid detection field, and with Nplex, this allows application like antisense oligo, as mentioned before, or SNP assays. These different formats will meet different requirements, but today I will focus on our validated Vplex and high sensitive Splex assays. Before I do that, I'll just mention our assays are built with the different stages of drug development process in mind. Here is our assay development process aligned with the phases from very early proof of concept, feasibility, and biomarker identification through the different clinical phases. This is, of course, a generalization, and all our assays has potential to be used in clinical trials, but they will require more or less work in the validation process from the CRO. Likewise, we have many users that use our validated assay in the early stage if they are already established. I will not go into details on the assay development, but if we unfold a little, you will see that we follow the phase-gated process and FDA guidelines for fit-for-purpose assay. It means that every assay needs to go through all phases to become a validated assay. Our validated markers, the Vplex and the Splex, are developed to actually bridge a gap between research and clinical biomarkers. In order to have an assay with high quality, with absolute concentration, rigorous quality, and enable high throughput and multiplex analysis. First, I will introduce the Vplex assay. The Rplex assay are validated assay. They are analytical validated, and they are bridged to ensure a lot-to-lot reproducibility and to meet requirements for longitudinal studies with few samples or larger phase 1 for phase 4 studies with maybe tens of thousands of samples. They come in large fit-for-purpose panels, but as you will see, they can be customized with selected biomarkers. The CV is typically low, and assays are normally off-the-shelf product that normally can be delivered in short notice. The development of products follow, as mentioned, a phase-gated process and a critical reagent program where all incoming reagents are qualified and assay tested against internal reference standards. Validation include multi-lot validation, analytical validation, and we provide a certificate of analysis for each lot produced. The assay follow a strict process that include that calibrators are validated against both internal and, if available, also external standards like WHO standards. For example, our latest for vaccine production has also been validated against WHO standards. So you can compare studies around the world and between different platforms. Of course, low CV and a verification of lower limit of quantification and upper limit of quantification for all kit lots. Specificity, recovery, and linearity for multiple sample types. Here's just an example of a 30-plex assay for optimized performance. They will be separated on 3 plates with assigned calibrators and diluents optimized to run these specific set of assays. Capture antibodies are pre-coded and detection antibodies delivered individually. And that gives us a benefit because if not all assays are needed, we can provide the very same panel in a custom mode where we only deliver the detection antibody that is requested in a specific multiplex. For example, here shown 5 selected biomarkers. They will be delivered on 2 plates, but we will only deliver 5 detection antibody, and you will maintain the validation of an assay. Same performance when we do multiplex and singleplex assays. And here's a correlation that show IL-2 and the correlation for an individual assay and IL-2 in a multiplex setting. Correlation data for all our assays are included in the protocol. And for long-term studies, longitudinal studies, as mentioned before, we show that we have a very robust assay platform over time. This graph shows a test of 78 independent lot over a period of time of 3.5 years. And this could be expanded because this has been product for many more years. And also the associated intra-plate concentration CVs, as you can see, is in the lower end. This Rplex product is used in many clinical trials, but sometimes we get requests on specific markers for a more high-sensitive platform. And for that purpose, we have Splex. The Splex is recently launched, but actually it has been used in pharma for the last 5 years, at least here in Denmark. But now we also have validated a few markers to launch to market, and they are validated very similar to the Rplex assays with increased sensitivity. Here again, TNF-alpha is shown, and why do we have an assay for TNF-alpha? I just told you before that the Rplex assay also measure TNF-alpha in normal serum samples. It is because it is towards the lower limit of detection. In the Splex assay, TNF-alpha concentration in normal samples will go into the quantitative range of an assay. Here also shown for additional markers from the pro-inflammatory panel, where you can see some of the challenging markers now with IL-2, IL-4, IL-6, and so on. Data will move in well into the quantitative range of an assay. That is a benefit when you look at small changes in a biomarker. Also, it is a benefit if you have precious samples, because you can then dilute much more and get more out of your sample. Also, some biomarkers is known to be below detection, and with Splex, it's now possible to measure those. And then we also see that for some assays, we can measure them in multiple matrices. An example is, for example, TSLP that we have launched recently, measured both in serum and, for example, sputum samples. The assay is designed as a sandwich ELISA assay, so it is still just coding a capture antibody, adding the sample, adding a detection antibody, and then the Splex has an extra enhancement of the signal, providing these extra levels of sensitivity. They are aligned with our Rplex assays, and there's a good correlation between them. But as you can see here on the left, this is now IL-10 and IL-17A. You can see that the Uplex, the Vplex with the blue line, is closer to the level limit of detection. And then when we move into Splex, we go further away from lower limit of detection and lower limit of quantification, as also shown on this graph. And here we see the correlation between the two platforms. Looking ahead, we will provide this Splex also in a multiplex setting. For now, it is a singleplex assay, but later this year, we expect to have also capability to run Splex in multiplex setting. I've mentioned that the MSD platform is relevant also in other areas, and for example, for therapeutic proteins, this slide show some of the application where the platform is used. For example, immunogenicity, a huge area, and now I just before told you about direct format. Also, a lot of immunogenicity assays is set up with a bridge format, and that has other advantages. I'll not go into further details, but I'll just mention these areas. If you, for example, also could be interested in, you're always welcome to reach out to us and discuss further. And then, yeah, of course, in this setting, we do collaborate directly with customers that do have our platform. We build assays in collaboration with our customers directly, but we also support transfer to CRO, in this case, for example, Unilabs, and we provide support with also our application scientists to implement the technology or to implement a specific study. And just in summary, before I hand over the word again to you, Dale, the platform, it is compatible with complex matrices. It is capable of multiplexing in a broad dynamic range with a high sensitivity. Now we have seen the different levels of sensitivity that different assays can provide. And we have a long list of assays. It is a rapid and robust operation, 90 seconds per plate. There's no fluidics, no calibration, and therefore also very little maintenance. It is validated for GLP validation. And then we have, yeah, a strong local support, and we're happy to discuss any project that you have. And with that, I will say thank you and give the word back to you, Dale.
Thank you very much, Anders. That was great. What can I say? That's a very impressive technology platform, and looks like the biomarker analysis capabilities are almost limitless, and the list is still growing. For everyone who's listening, please remember that if you have any questions, put them into the question and answer format down the bottom, and there'll be time at the end where we can ask our speakers the questions. Now I'd like to move across to our second speaker for the day. And I should say it's my great pleasure to introduce to you Dr. Colin Farrell. Colin has over 30 years of developing immunoassays for both clinical trials and other applications. He's worked in large pharma, he's worked in large CRO, and we're lucky enough now have him as the head of immunoassays at Unilabs York Bioanalytical Solutions Laboratories. Colin today will share with you now some of his experience from his 30-odd years of developing and conducting immunoassays. Colin, I'll leave it over to you.
Okay, thank you very much for that introduction, Dale. Okay, good morning, everybody. I'll this morning be talking about practical considerations for biomarker validation in support of clinical trials. And it won't be exclusively about the MSD platform. Hopefully, I'll share some thoughts on all platforms and the challenges that are typical to us, regardless of the platform that we're working on. So first of all, I'd just like to start with a definition of a biomarker, because I realise the audience may be varied for this presentation. So a biomarker is defined as a characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic interventions. Now, we're a CRO supporting regulatory bioanalysis for the pharmaceutical industry. So that's the area that we're mostly interested. A biomarker can be pharmacodynamic, predictive, prognostic, diagnostic, monitoring, a risk biomarker, or a safety biomarker. Now, the majority of the work that we do is for pharmacodynamic biomarkers. I would recommend, before you embark on any validation of a biomarker, is to understand the biomarker that you are validating And I must stress here, it's the assay that is being validated, not the biomarker, because there is a difference, and I would point you towards the Critical Path Institute paper that defines that very well. But all throughout this talk, I'll be talking about the validation of an assay for a biomarker. So firstly, how and what the data will be used for. So i.e. the context of use. And this is a very hot area in biomarker analysis at the moment. And I won't talk too much about context of use, but I'd point you towards the workshops that the EBF have, and also the Critical Path Institute. The aim of this talk is to get some practical considerations and the challenges that you'll have during the validation of the assay. So also, I'd recommend that you understand the mechanism of action of the biomarker, the anticipated levels in the target population, and also the anticipated magnitude of change in response to the drug. So the requirements of your assay would be very much different if you were expecting a 100-fold increase in biomarker in response to the drug, to an assay that you would be looking for a very small percentage decrease. The challenges are going to be somewhat different, and you need to adjust accordingly. Also, try and find out beforehand as much as you can about the stability of the biomarker, both in vivo and ex vivo. This may be that your biomarker is unstable immediately after collection, and that it may be a requirement to use enzyme inhibitors or preservatives during that collection, because you don't want a situation where you've got an unstable biomarker that is degrading as soon as you've collected it. It would make all the analysis of limited value if that's the case. Okay, in the next slide, I'm going to talk about assay availability for the biomarker. So first of all, do some research on what's out there and is commercially available. So as Anders has said, many of the biomarkers will be available on the MSD platform. And if they're not, it is an open system, and you can tailor it with the Rplex system that they have. But it may be that there are commercially available kits that are on the market that aren't for MSD, and you could get one off the shelf. So there are many ELISA manufacturers that will provide kits, and also there are other platforms. And that is the next question of what platform can be used. So we're not just talking about MSD or ELISA. There's the Ella and the Singulex platforms out there, as well as others. So if a kit isn't available, you then need to look at what reagents are available, and do your homework and try and ascertain which is going to be the best reagent for yourselves. Understand what assay range is required and compare that to what the kit might be offering, and that may decide on your choice of kit. Next question to be posing to yourselves is, what are the endogenous levels in commercially available control matrix? So you're going to have to make some QC and validation sample material, and to do that, you're going to have to use control matrix to do that, and find out what is available before you start. Also do research into finding out what analytical standards are available. So it may be that even if you've got a kit that that would be provided in sufficiently high concentrations to prepare your calibration standards, but not enough to prepare the quality control and other validation samples. So it may be that you have to source another standard, and this is likely to be a synthetic or recombinant form of the biomarker. And I'll discuss some of the challenges that they pose a bit later on in the talk. The next thing to consider is which of the validation parameters below that you're going to include in your validation. So this will depend on the purpose of the assay and whether you wanted to show that it is fit for that purpose. And it may be that you need to include all of these or just a few of them. So these would include the analytical measurement range, precision and accuracy, very important, and I would recommend this is always include parallelism, dilution linearity may be relevant. Also look at selectivity, specificity, stability of a frozen sample, out on the bench at ambient temperature, and also freeze-thaw stability. So all the challenges that your sample is going to be put through, you need to be replicating in your quality control sample, and you need to be demonstrating stability during the validation. Also, drug interference and interference of other co-administered drugs, or similarly, and similar endogenous compounds may also be investigated during the validation. As I've said, the quality control samples often pose the biggest challenge during a validation. And first of all, you want to screen the control matrix that you've brought in to do the study or collected to determine the endogenous levels. So if your assay is to support a clinical trial of human plasma, then the control matrix should be in human plasma. And ideally, you would screen the sources, individuals, pools that you have available, and it would cover the analytical range that you're looking for. So all the QC levels at the LLOQ, lower limit of quantification, the low, medium, and high levels, as well as the upper limit of quantification. And ideally, these would be all gained through endogenous levels, but that's not always possible. And if that's not possible, what do you do to obtain those different levels? So to get the higher levels, it's often necessary to spike that control matrix with a standard. Now that could be the standard that comes with the kit, or it may be an external standard that you've had to buy in a higher concentration from another supplier. Another option may be that you can stimulate whole blood to elevate the levels to the concentrations that you require for the higher QCs, or it may be necessary to dilute the endogenous control matrix with a surrogate matrix to get down to the lower levels. The MSD platform obviously offers multi-analyte options, and this poses challenge of preparing a QC that ideally is suitable for all of those analytes. But this isn't always going to be possible, that you may just have levels that you can't obtain across all of those analytes. So an option there is to have, for example, a low QC that is suitable for 7 or 8 of those biomarkers, but have a separate one for the remaining biomarkers that you would have in your panel. So just be flexible and use the best quality material that you can, that's going to be the most appropriate for the assay that you're developing and then validating. So the next slide, we're talking about the preparation of spiked quality control samples. So this is a scenario where you've not been able to obtain endogenous material at the higher levels, and also where you're spiking with an external standard. So you need to firstly understand how that external standard is going to behave in comparison to the kit calibration standards that have come with your kit. And to do this, I would recommend that you perform multiple calibration curves of that various material on the same plate. So prepare them at the same equivalent concentrations and assess what the performance of the external material is in comparison to the kit standards. And it may be that they are very similar, if not the same, in which case you can use the concentrations as they are. If they are different, then you need to adjust accordingly to obtain the QC levels that you require. And I just point out here that endogenous biomarker and spike synthetic recombinant standard may not always behave in an additive manner. So I would find out as much as possible in development of the assay before you move into validation, so you understand your biomarker and it in relation to it with this spiked analytical standard. And in the next slide, I'm going to demonstrate what I've just talked about there. So on the plate, you would be monitoring your kit standard, and that's the calibration curve that's obtained. So we've got assay response against concentration along the bottom. So on the same plate, you would test another analytical standard. And as I said, it may be superimposable, in which case there's no need to adjust your spiking regime. It would be as if you were spiking with the calibration standard. However, you may see that the analytical standard here, shown on the green line, behaves considerably differently to your kit standard. And in this example, we've got a parallel line running below. So regardless of the concentration you're spiking with, it's always a proportion below what you were actually aiming for. So here you'd apply a correction factor and spike with amounts that you have been adjusted for that correction factor. However, as shown with the blue line there, for the third scenario, it may behave completely differently across the analytical range, and you would have to tailor your preparation of QC or validation material across the board. So there's an element of trial and error in this, and again, iron out these problems in development so that you understand exactly how your compound is going to behave in validation. Once you've got the QC material at the levels, 5 levels that you might be validating over, you need to then do what we would call target setting. So at this point, you don't know what the nominal concentrations are. You've got an idea of what area of the analytical range they're going to be. But to ascertain the nominal concentration, I would recommend that you do multiple batches, at least 3 of them, and containing all the QC levels in replicates of at least 3, if not more. And assign these concentrations of the QCs based on the interbatch mean. Once you've got this interbatch mean, you can then retrospectively apply accuracy acceptance criteria to each of those intrabatches that have been applied, and that would be used as a nominal moving forward. And you can use your accuracy acceptance criteria, because prior to knowing that, all you can really do is test the method for its precision. The next slide, I'm talking about resource management, and I can't recommend this highly enough is that ideally you would prepare sufficient QC material for your validation, sample analysis, and any long-term stability. And as well as that, also purchase sufficient kits or reagents for the lifetime of that assay. This will avoid any lot-to-lot variation. Now, Anders spoke about the Rplex products that MSD have, and I would endorse what he said in that this makes bridging from lot to lot very much easier, but you might not be using those products, and a lot of the ELISA kits, the QC testing that is done is variable, and this is something that you would like to avoid. However, it's not always possible to avoid this. This may be because the sample numbers in a clinical trial you're supporting increase, or it may be that there's limitations on the production run that a manufacturer can do, and it's never going to be possible for you to secure enough of a particular product in one lot. And also it may be that the clinical trial runs over a long period of time, and that the kits or the reagents expire during that process, and you're forced into changing into a different lot. So what do you do when that lot changes? Either during the validation or in sample analysis. And we would recommend that you do a qualification batch as a minimum, and this would contain a set of calibration standards and also QCs at multiple levels in replicates of, again, at least 3. And ideally, it would meet the acceptance criteria that you've defined in your validation, and moving forward in your sample analysis, you've used that for each of the batches. However, if it's not met, there are a number of options that may be applicable. This could be that you reassign this the nominal concentration of the QC based on the lot of kit that you're using. It may have changed significantly, and that's the preferred route. Or it may be that you apply a correction factor to adjust the concentration you obtain to what you were getting previously with the lot that was validated. Or in the worst case scenario, it may be that you can no longer report concentration data, and that you would have to report sample data as a ratio. And this ratio is based on the baseline, so i.e. the pre-dose sample. And in the next slide, I'll show in a bit more detail how that would come about. So for this, I'd always recommend that you analyze a full profile for a subject in one particular batch, and you would still be using a calibration standard. So in the second column, we're getting concentration data out of that. But as I said, in this scenario, we're not reporting quantitative concentration data, but rather as a ratio. So that ratio is calculated relative to the pre-dose sample. So it's important that that pre-dose sample is always in the batch that you're analyzing. So here in the third column, I'm showing the ratio to that pre-dose sample, but you could see it may be that you get a sample that doesn't meet the sample acceptance criteria for your run, and no result is reported, and that's what we've got for the 180-minute sample. So that would need to be repeated. When you repeat it, you're going to have to repeat it with the pre-dose sample. So that in itself creates challenges, in that you need sufficient volume collected of that pre-dose sample to allow you to put that pre-dose into multiple batches if necessary. And also ideally that would be aliquoted at the point of collection, so that you're not having to put it through multiple freeze stores. So once you're analysing that in a separate batch, you get the result, and again you can calculate it as a ratio. In this slide, I just simply want to point out that biomarker assays are not the same as PK assays. The purpose of a PK assay remains the same, whereas a biomarker will the purpose of it might be different, and it might be different throughout the lifetime of that biomarker. It may be for different clinical trials, the importance of it changes, and that means you have to do additional validation work. So the requirements aren't less stringent for a biomarker, they're just different. And I would recommend that you treat them differently to the challenges that you would have for a PK assay. On the final slide, I'd just like to point you towards some guidance documents. There are many out there, and this isn't an exhaustive list, but the Lee paper from 2006 was one of the early ones to introduce the fit for purpose concept. And then the other two slides are documents are more recent. The EBF update paper from 2020 is an update on their thoughts on bringing context of use into practice. And also this is explored in the Critical Path Institute paper, which I would highly recommend as well. And that's the end of my presentation. So I'd just like to thank everyone for listening, and I'll hand back to Dale.
Great, thank you very much. First, I'd like to thank both Colin and Annas for giving such a great, insightful seminars, both into the MSD technology, but also what do you need to think about when you have a clinical biomarker in a clinical trial that you are investigating. There's time for questions here at the moment. If anyone has any questions, can you please put them in the Q&A function at the bottom of the screen? And we have a question here, which I believe would be for Colin to start with. If you have a marker where the values are below the limit of detection, can these still be used in statistical analysis, or how should they be treated?
I don't think you can include them if they're below the range that you've validated. I think you've got no option other than to report it as LLOQ. It may be that you can reconfigure your assay and do some extra, once you've established that the sensitivity requirements that you validate are greater than what you've validated, you could do some additional work to try and push the assay a bit further down in concentration. So, but if that isn't possible, I think you've got no option other than to report it as less than LOQ.
Yeah, and I can also comment that I agree with you, Colin. That said, we have customers, and maybe also to understand the MSD readout. So the lower limit of detection is based on the background, and 2.5% CV is determining the lower limit of detection. But we will give a count below lower limit of detection, and some use half a value of lower limit of detection to calculate statistically. But yeah, I will encourage you to talk to a statistician about this as well. Also, in regard to this question, there's lower limit of detection, and then there's lower limit of quantification. Lower limit of quantification in an MSD assay is set by us, but it is also possible to set your own lower limit of quantification. Ours is conservative set, but yeah, but that is also a difference between the quantitative area and the detectable area.
Great. I have one more question here actually for you, Annas. You presented the Vplex and the Uplex. you have some fantastic panels of everything you'd need for, say, for example, pro-inflammatory panel. If you're only interested in, say, 5 of the 10 markers on the panel, you said that it is possible to select. What would be the recommendation from MSD? Should you take the VPlex panel with only half, or would you suggest that you could do a Uplex panel where you mix and match your own?
Yeah, so it depends on the study. It depends, as Colin also explained in his very nice presentation, that you need to look into what are the dynamic range of an assay, what is the purpose of an assay. As I explained also, the Rplex assay is validated for later clinical trials and easier to use in clinical trials, but the Uplex is also used in clinical trials. And the benefit is that you can conserve sample because maybe all markers can be fit into one panel. So I think it will require a discussion about the matrix in question and also the markers we're talking about. We do not have a general recommendation on that.
Okay.
I hope that was a clear answer.
That was. So I have no more questions online. If anyone has a quick question, please write it. Otherwise, it's 1 minute past 11. I see. If there are any more questions from anyone in the audience and they don't want to put it up in this forum, please contact us at Unilabs Serving Pharma, and we can either get you in contact with Colin directly or Anna's, and you can have a more specific discussion about your specific project. On that note, I'd like to, again, thank both Colin and Anna's for presenting today and hand back over to David at Medicon Valley Alliance.
Thank you very much, Dale. And on behalf of Medicon Valley Alliance, it's just for me to say goodbye and hope to see you later at future MBA events. I hope this morning has been time well spent. And as mentioned earlier, if you're curious about this concept and other event concept and how you can work with MBA in that regard, don't hesitate to reach out to me. Of course, I would also like to on behalf of MBA, to thank the speakers and the entire Unilabs team who made this this webinar possible. And with that said, I think we've reached our destination, so to speak. And I will conclude this inspirational webinar and just say to all of you, take care out there.